I would like to begin by acknowledging that we are located on Treaty 1 territory, the ancestral lands and traditional territories of the Anishinaabe, Niniwak, Athinawak, Dene Sulin, Anishinaabe, Inuit, Dakota, and Nakotuz peoples, and the homeland of the Métis Nation. We also acknowledge that our water is sourced from Shoal Lake 40 First Nations. So I have the pleasure today to introduce Dr. Cheryl Greenberg and Angela Krutish, who will be presenting on inborn errors of metabolism and the OMICS First DNA testing. Dr. Greenberg completed her medical studies as well as her postgraduate specialty training in pediatrics and medical and biomedical genetics at McGill University in Montreal. She subsequently moved to Winnipeg in 1978, where she is proud to have lived ever since. Throughout her professional career at the University of Manitoba and the program of genetics and metabolism at Shared Health, she has focused on caring for patients and families with hereditary metabolic disorders, as well as taking a bedside to bench and back to bedside approach to further clinical discovery and find new and better ways to treat patients. Dr. Greenberg is the academic lead for the Canadian Prairie Metabolic Network research study. And Angela Krutish today will be speaking to us, is a board certified genetic counselor. She currently works at the University of Manitoba as the project manager for the Canadian Prairie Metabolic Network research study. And her primary, in her prior roles, Angela worked as a clinical genetic counselor in Thunder Bay, Ontario. Angela's main interest is finding ways to improve access to genetic services. Therefore, she's excited to be part of this CPMN team that we're going to be learning more about. In her spare time, Angela enjoys reading, cooking, and spending time with her family and 3 dogs, and I share those interests as well. So thank you both for coming so early in the morning and presenting to our group. I know there will be lots of families and clinicians that will be watching this recording at a later date. So thank you so much for joining us, and I will now turn the presentation over to you.
Good morning, everybody. Can everybody hear me? Okay, good. Thank you so much for inviting Angela and I to speak to you today about our Canadian Prairie Metabolic Network project called OMICS First DNA testing. Angela really is going to be doing this presentation. I'm just going to sit back and enjoy this. We've worked together now over almost a year now, in terms of beginning to implement this study, and we're very excited about it. I know I'm speaking to clinicians in the audience, various healthcare providers, doctors, nurses, therapists, engineers, pharmacists, quite a broad variety of healthcare providers and researchers. And I also know, if not right now, but certainly in the future, some of the patients, and their families, may be viewing this talk. So there's really quite a broad range of people. And I think we're very excited to begin to introduce the concept that you can order DNA testing first in the diagnostic workup of a patient suspected of having an inborn error of metabolism, as opposed to the diagnostic odyssey that many of these patients have gone on in the past. Now, for the patients in the audience, some of you with your children have been through diagnostic odysseys, and maybe some of you are just beginning. So if any of this talk resonates with the clinicians in the audience and the families, please do not hesitate to contact us, and we'll explain to you more about that to see if any of you think that your patients would be eligible for this program, which we're evaluating in parallel with the traditional routes of how we diagnose inborn errors of metabolism. So I'm going to say a few words at the end again, but without further ado, I'd like to ask Angela to take this over. Angela?
Perfect. Thank you so much, Elizabeth, for introducing us, and Dr. Greenberg for that introduction to the talk. So I will get right into it. We've had a land acknowledgement already. Just briefly here are our disclosures. Dr. Greenberg's are listed there, and I myself do not have any conflicts of interest to disclose today. So these are the learning objectives that were circulated ahead of time. So just to run through them briefly, after this talk, I hope that you will all be able to define inborn errors of metabolism and list some of the common signs and symptoms of these conditions, especially in the pediatric population. Recognize different types of genetic testing, such as single gene testing, multi-gene panels, whole exome sequencing, and whole genome sequencing. Be able to contrast the OMICS First approach, which I'll tell you more about, with the traditional approach to diagnosing inborn errors of metabolism. And last, to consider the potential benefits, drawbacks, and expectations of this OMICS First approach used for the diagnostic workup of a wide range of genetic conditions, but primarily I'm going to focus on the inborn errors of metabolism. So as you can probably gather from the learning objectives for the talk today, I'm first going to talk about inborn errors of metabolism, then kind of do a bit of a genomics 101 to make sure we're all on the same page and talk about the evolution of genetic testing over the years. And then last, give a little bit of an overview of this OMICS First approach that we're using in the Canadian Prairie Metabolic Network study. So to start off, inborn errors of metabolism, or IEMs, are genetic conditions that really affect the way the body breaks down food, stores it, and turns it into energy. So there are many different groups of IEMs, but some of the main groups we think of include disorders of carbohydrate metabolism, fatty acid metabolism, protein metabolism, storage, and the production of energy. And so this diagram kind of shows what we generally see with inborn errors of metabolism. So there's some sort of substrate or something that's coming into the body that's broken down, and that's what typically happens. But in the person who has an inborn error of metabolism, there's a problem in that pathway, and so that substrate can't be broken down properly, which leads to an accumulation of the other things that substrate is generally broken down into, and the lack of the product that we would normally be getting. So that kind of gives you a general scheme of what happens. And it's really those effects that tend to cause the signs and symptoms that we see in inborn errors of metabolism. So to give you just an appreciation of the many different metabolic pathways at work in our body is this diagram that shows a bunch of the different pathways. And so I think just what I hope you can appreciate from this is that there's a lot of different pathways going on, and there's a lot of places where something can go wrong. So individually, these conditions are rare because you can see all the different places that an inborn error of metabolism could happen. But what I want to make clear is that collectively, these conditions are actually quite common. And so it's thought that there could be as many as 1 in 800 people in the population who have a metabolic condition. So as the name suggests, inborn errors of metabolism are something that people are born with. And that's because they're caused by a change in the genetic information, which I'll explain a little bit more in the later slides. So classically, inborn errors of metabolism were thought of as being childhood onset, with the first symptoms occurring very early in life. But what we're actually learning is that inborn errors of metabolism can present throughout the life cycle, where some people may develop their first symptoms later in childhood, in adulthood, or even into their senior years. And so in fact, about 50% of patients with inborn errors of metabolism have the symptom onset at later than 1 year of age. So as you can see, about half of patients don't actually have any symptoms when they're an infant and very early in life. And so this slide here is a list of signs and symptoms that a healthcare provider may encounter in a patient that should make them think of an inborn error of metabolism. So this list is targeted a little bit more towards what you might see in the pediatric population. And I've highlighted and bolded some of the ones that might be especially relevant for this audience. And so for those that aren't clinicians, the signs and symptoms usually involve the central nervous system, and they can actually sometimes appear suddenly in somebody who was previously well, whether that be a child or an adult. And so for those of us that are not seeing patients, what typically happens is when a patient is referred to a specialist, such as a geneticist or a metabolic specialist, the healthcare provider assesses the signs and symptoms, creates a list of possible diagnoses that this person might have, and then they might order many different blood and urine tests to try and sort out what is going on for that person and what is the cause of their symptoms. So in the research study we're going to discuss here, we're asking that instead of creating a list of possible diagnoses or tests, healthcare providers take a more holistic approach, think this might be metabolic, and then consider referring the patient to the research study that I'm going to tell you more about. So I just want to talk a little bit about the different ways that an inborn error of metabolism might be picked up or diagnosed. So first is on the newborn screening test, which is also called the heel prick test. So this is a screening test that's done about 24 to 48 hours after birth, and it screens for more than 40 different rare conditions. Most of the tests are done using a small amount of blood from the baby's heel, which is why it's called the heel prick test. It's only a screening test, but it's a way of picking up children with some of the classic inborn errors of metabolism for which there are known effective treatments. So it's really important to pick them up early so that we can start treatment as soon as possible for those children to make sure they have the best outcomes possible. So what about for conditions that aren't picked up at birth? So as I've kind of alluded to, clinicians might order metabolite testing. And so essentially these are chemical tests that are often done on blood and urine samples, but they can be done on different samples or tissues from the body. And so this is really, when we think back to those metabolic pathways, it's looking at different parts of the pathway to try and find out, is a level higher than what we normally see, is something missing, and really to try and sort out what pathway might we be suspecting there's a problem in. The other type of testing we can do for inborn errors of metabolism is molecular or genetic testing, also called DNA testing. And I'm going to tell you a little bit more about that in the coming slides. And so together, the metabolite testing and the molecular genetic testing is what I'm really going to focus on today. I'm not going to talk much more about the newborn screening. Okay, so like I said, I'll give you a little bit of a history about genetics. So in 1956, scientists confirmed that the human cells have 46 chromosomes, which are separated into 23 pairs. So we have 2 copies of every chromosome. One comes from mom, one comes from dad. So in this figure on the right, you can see the first 22 pairs. It doesn't show the last pair, which is the sex chromosomes. So you may have heard of those before. People that typically have 2 X chromosomes are considered to be biologically female, and those that have 1 X and 1 Y are biologically male, although we certainly can see some differences in the sex chromosomes. Now here's kind of a diagram of what does the genetic information look like in a cartoon form. So all of our cells contain a copy of the genetic information, which is separated into these chromosomes. And those chromosomes are actually a very, very long piece of DNA that's all wrapped up. And so the 23 chromosomes, so 1 from each pair, actually contain a lot of DNA. The DNA is broken into letters. So kind of like the English alphabet, but there's only 4 letters, A, T, G, and C in the DNA alphabet. And so that DNA is all wound up about 3 billion letters into one whole set of chromosomes. So you can appreciate that's a lot of information. And the chromosomes together encode about 20,000 pairs of genes. And so the genes are like the blueprint for how our body grows and develops and functions. So at first we learned about the chromosomes, and then the techniques that scientists use to study chromosomes evolved into the study of the DNA. So in 1990, a project called the Human Genome Project was launched in the US, and the goal of that was to sequence the human genome. And so what I mean by that is to go along all the DNA and read all of those letters. And so that project was completed in 2003 when they had sequenced about 92% of the genome. There are some parts of the genome that are harder to read, and so those were the pieces that were missing. But some of you may have seen an article. There was a lot of news about it just a couple weeks ago that the genome has now been completely sequenced for the first time. So they were able to go back and fill in that 8% that we didn't previously know the sequence of. So that was a really exciting breakthrough this year. So how do we actually look at the genetic information? And why is this important? So it was really important for us to learn about genetics and what our genetic information should look like. And so that we would be able to tell then when is there a problem with the genetic information. And that's important for diagnosing genetic conditions. So the first thing that we can see, or that I'm going to talk about here, is how are different genetic conditions caused. So they can happen in different ways and due to different problems in the genetic information. I'm not going to cover everything, but I'm just going to cover a few things that clinicians may be familiar with, patients may or may not be familiar with here. So the first thing we can do is we can actually look at those chromosomes. We know that people generally have 46 chromosomes, like I said, but sometimes people can have an irregular number of chromosomes. So for example, people in the audience may be familiar with Down syndrome, which happens when somebody has an extra copy of this chromosome 21. So they actually have 47 chromosomes instead of 46. Another way that a genetic condition can be caused is when there's an irregular structure of the chromosomes. So you can see we know what the chromosomes generally look like, but sometimes people may have extra or missing pieces of chromosomes, or pieces of the chromosomes may be in the wrong place. And so a deletion happens when someone's missing a chunk of a chromosome, or a duplication can happen when someone has an extra chunk of a chromosome. And so that can sometimes cause genetic conditions as well. Now, another way of doing genetic testing is to actually go and read along all the letters of the DNA. And we're looking for an irregularity in the DNA sequence. So we're looking for a letter in the wrong place, a different letter than should be there. And that's what we call a mutation or a variant in the DNA. And this is going to be important when I talk a little bit more about this research study. So you might be wondering, okay, what testing do we use for inborn errors of metabolism? And to understand this, it's really important to understand the cause of inborn errors of metabolism. So inborn errors of metabolism are generally caused by a single gene, and they're typically inherited in what we call an autosomal recessive pattern, which is shown in this diagram here. So because we all have 2 copies of every chromosome, this means we also have 2 copies of every gene. And 1 copy comes from mom and 1 copy comes from dad. So when somebody has an inborn error of metabolism, it usually happens because they have a problem or a mutation in both copies of a gene. So as you can see in this figure here, if we have a dad who has a mutation in 1 copy of the gene, but the other copy is unaffected, so working normally, and mom has the same thing, a problem in that same gene, and then another copy that's working normally. If both parents pass the copy of the gene that carries a mutation to their child, then that is how an inborn error of metabolism happens. And so for this reason, for inborn errors of metabolism, we usually do tests that read along the DNA and look for these mutations, which I kind of like to think of as they're like a spelling mistake in the DNA. So as our knowledge of genetics has grown, the genetic testing technologies have also advanced. So initially, when we were able to read the sequence of DNA, we were usually looking at single genes. So this isn't to say that single gene tests aren't still useful. They're still used if we're highly suspicious of one disorder. So just to throw an example out there, if we think somebody has Huntington disease, we would just look at a single gene. As our ability to test for more and more things grew, multi-gene panels were developed. And so a multi-gene panel usually looks at many genes that are associated with a specific symptom. Again, these tests are often still used if a patient has a very specific presentation. So for example, a patient who has seizures may have a panel that looks at genes that we know can be associated with seizures. We now have access to more comprehensive tests as well that look at all the genes at one time. And these are called whole exome and whole genome sequencing. Now, I know this is getting a little bit technical and I didn't give all the information about this before, but just to give you a little bit of an understanding of the difference between exome and genome sequencing, both look at all of the genes in the body, but the difference is that the exome only looks at the exons in the genes, which are the parts of the genes that make proteins. So the parts of the genes that make things that go out and do jobs in the body. And so the exome is actually only about 1% of our genetic information, but it's really the part that we understand the best right now. And so that's why whole exome sequencing is kind of the biggest test that we generally are using in the clinical setting right now. More and more in the research setting, especially, though, we're starting to see the use of whole genome sequencing. And that's really looking at, as you can see, the whole apple. It's looking at the sequence of all of the genetic information. So looking at all of those 6 billion DNA letters. So now that you have a bit of a background about genetic testing, I'm going to go back to this slide about how do we look for inborn errors of metabolism. And so again, we're going to focus on those that are not screened for at birth. And those are typically worked up using what we're going to call the traditional approach, which is a combination of this metabolite testing, so the chemical tests in blood and urine, and molecular or genetic testing. Now, genetic testing often will start with single gene or multi-gene panels, and then might progress to whole exome sequencing if the prior tests are unrevealing. So this is how we're typically doing things in the clinic right now when we think a patient has an inborn error of metabolism. So as you can probably imagine, maybe you've experienced yourself if you're a patient or a family member, or if you're a specialist, maybe you've probably seen this in many of your patients. This entire process can take a very long time, often several years, which is why it's often referred to as a diagnostic odyssey. It's, as we know, it's taxing on the patients, on the families, and also on the healthcare system as well. And so this figure just kind of represents some of the pieces of why it's really an odyssey. So we see that on average, patients that have a rare disease may see 3 to 5 specialists, and 10% of those will see more than 11. About 40% of them receive 3 or more misdiagnoses. It generally takes, as I said, years to get a diagnosis, and about 30% of those people still remain undiagnosed, with nobody really able to find an explanation for their symptoms. So that's where our study comes in. So the OMICS First approach is proposed as an alternative to the traditional approach for working up suspected inborn errors of metabolism. So instead of the traditional path of the diagnostic odyssey, including wait times to see a specialist and the traditional approach of that metabolite testing combined with genetic testing, the OMICS First approach goes straight to comprehensive DNA testing through whole exome or whole genome sequencing, which was the DNA test they talked about that looks at all of the genes at one time. So this slide just kind of gives you a contrast of the two approaches. And I think what's important to understand is that right now, this is a research study. And so we're trying to compare the OMICS First approach to what's traditionally being done. And so these two approaches are being done in parallel. So, to give you an idea and a better understanding of what does a traditional approach usually look like for patients suspected to have an inborn error of metabolism, depending on their age, the wait list to be seen in the metabolic service may be between about 0 to 2 years. Once they're seen in the metabolic service, and let's say they're going to have genetic testing, the test approval generally takes about 3 to 4 months. It goes to this out-of-center committee, which decides if they're going to approve that testing or not. If the test is approved, the patient then has to have usually a blood sample drawn for the testing. And then once the testing is done, it usually takes about 4 to 6 months until the results come back and the family receives those results. So overall, that can take about 1 to 3 years. So you can see where that diagnostic odyssey really starts to come in. The other piece that's missing here is I didn't include the piece about the metabolite testing. So sometimes that may come in, before we decide to do a genetic test. we may do a genetic test that's negative and then do some more metabolite testing. So this timeline can really vary, but it's just kind of to give you an idea of how long that process can really be. So with the OMICS First approach, what we're aiming to do is consent a patient and take them to the triage meeting, bring their case to the triage meeting within about 2 weeks of when they're consented. For those that are approved for the study, we collect the sample just by a cheek swab instead of a blood test. And then the testing generally takes about 2 to 4 weeks to come back, and then we provide the results to the family. So overall, we're kind of aiming for the whole process to take about 4 to 6 weeks. That said, there's been some logistical challenges, which I can certainly comment on if anyone has any questions. But I think what you can appreciate from this is that this approach is quite a bit faster than the traditional approach. So because we're starting the study with comprehensive DNA testing and offering it in a rapid fashion, our hypothesis is that the OMICS First approach will lead to faster timelines, so patients getting things done faster, a higher diagnostic yield because we're doing that comprehensive DNA testing, fewer tests and investigations, so whether that might be imaging, invasive tests like muscle biopsies, things like that, fewer and shorter hospital stays, the same or lower cost to provide the care, and then on the other side, the same or better quality of care. So it's also important to make sure that patients are still receiving the type of care that they would like. So just to go into a little bit more detail about the study process for those patients that are referred. So when we receive a new referral, our research nurse contacts the patient to obtain informed consent. He then reviews their medical records to put together a really comprehensive summary of their medical history. So as you can imagine, a lot of these patients do have a complex medical history with a lot of different signs and symptoms that he reviews. The case is then brought to our weekly multidisciplinary triage meeting, and we kind of take a consensus decision-making approach to determine whether patients are eligible for the OMICS First testing. And I'll say, by and large, most patients have been approved for this testing so far. When patients are deemed eligible for testing, as I said, we collect a cheek swab sample for the DNA testing, and the cheek swab sample is then sent to a private lab in Calgary called Discovery DNA. Once the results are available, we again review them in that kind of team model at our variant meeting. And the results are then returned to the referrer and either provided to the patient by the person that made the referral or by our team. And generally it's been our team who's been providing the results to the families. And then we arrange any follow-up, which I'm going to talk a little bit more about in a couple slides. So now I just want to give you a couple case examples to kind of appreciate this OMICS First approach. So this first one is from the literature. And this was a 2-month-old male who was admitted with chronic diarrhea and failure to thrive. And he was transferred to the pediatric ICU with worsening metabolic acidosis, hypernatremic dehydration, and hypoglycemia. And so in this study, they were doing rapid whole exome sequencing on patients who had new onset or metabolic or neurological disorders. And so they did the exome sequencing, which showed a mutation in the PIK3CD gene, which is a gene that mutations in cause primary immunodeficiency. And so what I really want to focus on here is what were kind of the benefits and the outcomes of doing that OMICS First testing. So first, it was a very rapid timeline, as they had aimed to do. So the average time for this study team to get a verbal report from the lab was about 8 days, and they generally received a written report in 2 weeks. In the paper, they commented that they avoided doing some further invasive tests that they would have otherwise considered for this patient, which I would also speculate could have potentially saved some money as well. Importantly, the diagnosis guided the treatment. So they initiated IVIG infusions and made diet modifications, which were said to significantly improve the patient's symptoms. And then I think the last piece, which is really important and shouldn't be overlooked, is the impact this had for the family. So they commented that the family was pleased to have an answer for what was happening for their child. They were comforted by having an answer and felt comfortable going forward, not pursuing any additional invasive tests now that they had this answer. The study team was able to provide a bit more of a prognosis and kind of expectations for what the family could expect going forward with their child. And last, because there was this genetic information now, they were able to talk to the family about, you know, what is the chances this might happen again if they decide to have more children. So you can see that from kind of the medical system and the management perspective, it had a lot of impacts, but also from the psychosocial perspective for the family as well, which is something that we're also quite interested in for this study. Now I'm going to give you an example from our study. So that was one from the literature. Here's an example from our study. So there was a 6-month-old male who was seen for hyperexplexia, or an exaggerated startle response. And so I'm going to walk through the history here. In terms of the pregnancy and birth history, it was an unremarkable pregnancy. He was born to a 24-year-old mother at 38 weeks by spontaneous vaginal delivery. The APGAR scores were both 9 at 1 and 5 minutes. And the birth weight, length, and head circumference are here. You can see within the normal range. In terms of the baby's development, at 6 months old, he was sitting with support and starting to sit without support. He was reaching for objects and bringing his hands to the midline. He was fixing and following, and there were no signs of regression. On the right-hand side here, I've just included the family history. As we mentioned, the inborn errors of metabolism are caused by genetic changes. So of course, always important to take a family history. And so you can see in terms of the family history, this is the patient here with the arrow pointing to him. He had one sister who had bilateral clubbed feet, and he had a maternal great aunt who had seizures. But other than that, nothing too significant in the family history, though dad's family history was unknown as he was adopted. On his physical exam, when he was 6 months old, you can see that he was quite small. So his weight was under the 3rd percentile. His length was at the 15th percentile, and head circumference at the 5th percentile. He had mild hypospadias, and there was a question of whether there may be some early signs of developmental delay. He did have some workup to try and find out why he was having this exaggerated startle response. He had the metabolite testing, which was non-diagnostic, didn't really give us a clue as to what was going on. He had a microarray, which is the test that looks for extra or missing pieces of the chromosomes, and that was normal. He had an EEG looking for seizure activity, which was normal. And he had imaging of the brain as well, which was normal. So from the workup so far, really no clues as to what was going on. So some of the things that could be happening from a genetic perspective are hereditary hyperexplexia, glycine encephalopathy, which can cause the hyperexplexia, and that specifically is a metabolic condition or an inborn error of metabolism. And then there's also a range of other neurodevelopmental disorders that can have an excessive startle response. So he had whole exome sequencing through the study, which found a variant in the ADCY5 gene. Now, what I'm going to direct your attention to here is the classification. I didn't comment a lot about this, but when we do genetic testing, the first step is looking to see, is there a problem we can find in the genetic information? But it's not just as simple as that. The next step is then actually the lab needs to say, well, do we think this gene change is something that causes a disease or pathogenic? Or is it something that just makes this person different from everyone else? So we all have gene changes. They don't always cause problems. So that's really, I think, the difficulty for labs and for clinicians right now is not identifying the gene change, but actually saying, is this significant or not? And so a lot of the time we find a gene change that's called a variant of uncertain significance. And so how I usually explain that to patients is it's kind of like a big question mark. We don't really know whether it's pathogenic or disease causing or benign or something that's not disease causing. And so that was what was found for this boy. Now, one of the first steps we often take to try and resolve these variants of uncertain significance or get a better handle on whether we think they're suspicious or not is test the parents. If the parents do not have the same symptoms as the child, then we would not expect them to have that gene change if it was the cause of the child's symptoms. And so it can be very helpful to test parents. And so in this case, this gene change was found to come from dad. Dad was healthy, had no concerns overall. So this was thought probably not to be the cause for the baby's symptoms. So at the end of the day, you can see with this case here, we did this extensive testing, but there was still no molecular diagnosis. So let's just talk a little bit about what are the benefits and drawbacks of the OMICS First approach here. So in terms of the benefits, this may have limited additional tests. So especially in terms of metabolite and molecular testing. So there would definitely, I think, would not have been any additional genetic testing initiated after this. There was also a rapid turnaround time. So I think I have the time written down here. So it was 12 days from when the sample was received in the lab to when the results were reported out. So again, quite fast turnaround time, kind of on par with what we saw in that study from the literature. In terms of the drawbacks, there was still no answer. So, and I'll talk a little bit on a later slide about, kind of what do we expect? How often do we expect to find an answer? Because we certainly know that this testing is not going to find an answer for everyone. And then the other piece is that while the turnaround time for the initial testing is quite fast, the follow-up piece, such as doing parental studies or other investigations for these variants of uncertain significance, can actually be where the time starts to add up. And that's what we're kind of finding so far with our study and the additional investigations we're doing for a lot of different patients. So while the initial test may be fast, sometimes that follow-up piece is actually the piece that takes a lot of time. The other thing I just want to note here, because I talked about the metabolite testing a little bit, is it's important to note the metabolite testing is still critical. So in terms of proposing this OMICS First approach, we're not saying to do away with the metabolite testing. I think we're just saying that it may be, in these cases, more key kind of on the back end of things. So once we have the DNA testing back, the metabolite testing may be really helpful in a lot of the cases to try and really confirm the results that we found to see, okay, we see a problem in this gene. Do we actually see changes in the substrate and product levels? Now, this third one here, I'm not going to present the entire case, but I just wanted to share a quote from one of the patients. So the funding agency for this study is writing an article about the study. And so we asked a couple of patients if they would mind sharing their experience with the study. And so this patient here said, "I had a very positive experience with this study. It was a simple DNA cheek swab test that yielded results in a very short time. The research team made sure to answer any and all of my questions and kept me informed along the way. In any case, my results were the best case scenario. There was not enough significant evidence for genetics to pursue any more testing." She then says, "My brother has gone through years and years of genetic testing to get a diagnosis. Traditional genetic testing is incredibly expensive and time consuming, which makes me think, what if this would have been an option for him? I can't imagine how much time and cost savings to our healthcare system this could be, but more importantly, how much stress this would have eliminated for him and any other patients waiting months on end for each test result to come in, then only to start all over again with the next round of tests. This is where frustration sets in. I really hope this will help many others to get the answers they are looking for. There is nothing easy waiting for test results of any kind." So I think she really captures some of the things that we're hoping this OMICS First approach may provide for patients. So again, I just want to highlight this here. There's a lot of different things that we're evaluating and comparing between the OMICS First approach and the traditional approach to diagnosing inborn errors of metabolism. So we're looking at what proportion of patients are we finding a diagnosis for? Does it have changes for their treatment and management? What is the timing like? Is it really shortening that timeline? And then of course, also what are the costs and resources needed to do this OMICS First approach instead of the traditional approach? But the last piece, which I think that quote really exemplified, is that we can't overlook also the psychosocial impact, the impact on the families and the patients, while they're on this journey to try and find an explanation for their symptoms. So if you are a healthcare provider and you think that you have some patients that may be eligible for this study, that is excellent. The eligibility criteria is really, patients can be any age. They just need to be suspected to possibly have an inborn error of metabolism. And of course, they should consent to the referral. So please let them know you're going to refer them to the study so our nurse can get a hold of them. It's really important to note that any healthcare provider can refer to us. It doesn't matter whether the patient has already been seen or referred to or seen by genetics and metabolism. If you send them to us and they haven't been referred to metabolics, we'll make that referral because we really want patients to kind of do the two approaches in parallel. And so if you have a patient you would like to refer, I've highlighted our website at the bottom here, but certainly contact us as well and we can give you more information. Our referral form is available on the website. You can either download it and fill it out and send it in, or we actually have a fillable referral form on the website as well. So as I mentioned, I would talk a little bit about what do we expect with this study. So we expect that about 25% of patients will have a diagnosis, but many will not be classic inborn errors of metabolism. So because we're looking at all the genetic information, and we know that the symptoms that can be suggestive of inborn errors of metabolism are not always necessarily very specific. So we expect that some of these conditions we find will not be classic inborn errors of metabolism and in fact other genetic conditions. And then this picture of the road here, I think shows just the long road that we do still have ahead. So right now we're working on gathering the evidence about the OMICS First approach and building a clinical network. I didn't really talk about this, but we're really trying to build a network of metabolic specialists across the prairies. But going forward, if the province were to eventually adopt an OMICS First approach, there's still a lot of questions to be answered, such as who is going to have the time to do this? Who's going to be responsible? And what will the cost be and where will that funding come from? So I think I will turn it over to Dr. Greenberg right away. I'll leave it on the acknowledgement slide here. She may have something specific she wants to say about all the people involved.
Okay, thank you. Thank you, Angela, for an excellent overview. As you can see, and many of you know who do research, whether it's clinical research or basic research, research is team science. And, there's so many people whom we have to acknowledge that made this Genome Canada grant a reality for us. And obviously, I will not go through all of them, but it really has been a team effort. I wanted to just summarize few things that Angela has stressed throughout the talk. In many branches of medicine, everybody is moving towards using genomic diagnosis as a first tool to help establish a diagnosis and plan management of a patient. But establishing a possible genomic diagnosis is really only the beginning. And we'll see this throughout our CPMN project. First of all, Angela highlighted the difficulty in understanding and interpreting variants of unknown significance. That's a huge challenge in terms of interpreting the genetic information that comes back to us with the results. We need to move forward, regardless of how the study ends up, that what we find, if it really shows that a precision medicine, OMICS First approach is better for patients, we need to make sure that we have the right supports in place for our program in metabolic genetics to deliver the best genomic medicine in the future over the next 2-3 decades for our patients. And I hope that our study will be able to provide the evidence that we need the supports to help provide a new way of diagnosing inborn errors of metabolism along with the traditional. from this study, we're seeing that there are many other issues that come up. First of all, about issues related to data storage, sharing of data between the provinces, because it's not just Winnipeg that's a center for the CPMN, but we're bringing on Saskatchewan and Alberta, and there's many issues related to data storage, sharing of data, who owns the DNA. And of course, if you do make a diagnosis, what next? How can we translate this into participation in new clinical trials or access to new medications? So really, the CPMN is just the beginning of a whole new approach to how to incorporate precision medicine and genomic medicine into our everyday practice. And not just for metabolic genetics, but for every branch of medicine. And in the end, we just want to make sure that patient outcomes are improved, and that really is the ultimate goal. So I think we have time for questions, and we're happy to answer any questions you may have.